Okay, hello everyone, and I'm going to try and complete our discussion of DNA function in prokaryotes, and we'll see how well we do today. There may be a little bit left over for Monday. So I should explain, I've put up a new PowerPoint set to succeed what the previous one. And I am going to return to a topic that I really just glossed over, which is control of gene expression in the prokaryotes. I decided that I need to explain it in more detail for it to really be comprehensible, and it is a really important topic. So I remember what I told you about the nature of genes in general, universally. Not only in prokaryotes, but there are sets of genes which are read off the whole time. Messenger RNA is produced from those genes continuously because cells need their product continuously. But many other genes actually need to be controlled. They are only read off when they are absolutely necessary. And there are different ways in which the cell can control gene expression. We are going to discuss two kinds, which are specific to the prokaryotes. And these are one set of genes called the inducible genes, the other called the repressible genes. And you'll see the reason why in a minute. So repression is the actual inhibition of gene expression. And it is mediated by sets of proteins called the repressors. And in the repressive system, the default position of the gene is that it runs the whole time. It is on the whole time until a repressor is activated and stops the gene from working. So the opposite is an inducible gene, and in an inducible gene, induction turns on gene expression, as you guess, and it is initiated by something called an inducer, the nature of which we'll see. The default position for an inducible gene is that it's off. It stays off the whole time until it is induced to start operating, being read to produce the messenger RNA. So this will become clear. I just want to remind you of what we said about the structure of the operon. Operons are specific to the prokaryotes, and they're an organization of related genes. So instead of having genes scattered all over the genome, instead, genes which are related to a particular function are grouped together so that they can be controlled by a single promoter. So this is what an operon consists of. First of all, we have the genes of interest here. They're grouped together, and they will be read, the messenger RNA will be read off as a single strand, which is then cut to produce the working strand. Upstream of the working genes, there is a switch area. It's called the operator, and it is a switch of some sort. Above that is the promoter, and you'll remember the promoter is where RNA polymerase binds initially before it begins transcription of the genes into messenger RNA. So that is the operon, promoter, the operator, and then the structural genes, whatever they are. Somewhere else in the genome, here it's shown being adjacent to the operon. It does not have to be adjacent to the operon. It depends on which operon you're talking about. There is somewhere a gene which is going to produce the regulatory protein, and we'll see the different ways in which it can work. So here we're talking about an inducible operon. The default is that this gene remains off. It remains off until a signal is given for transcription to begin. So this is a very, very famous operon. And I'm sorry. and it's called the lac operon. And in fact, it was one of the first operons to be deduced, let's say. And so the lac operon, it operates to catabolize lactose. So lactose is a sugar. It's the sugar that is found in milk. Bacteria are not necessarily exposed to lactose very often, but there are some which are able to metabolize it. And this is especially important in the all of those bacteria that are associated with the gut. So this is one of the reasons why, for instance, in lab, you've been testing for lactose, testing to see whether organisms can utilize lactose, because it's a very important indicator of those enterobacteria like E. coli, those, the enteric bacteria. So here are the genes. They are the gene names are Z, Y, and A. Here they are here, and they lie right next to one another. Just upstream of them, here is the operator there, and here's the promoter, and here's the inducer here. The gene for the inducer is here, somewhere else on the genome. That runs continuously. That gene runs continuously and is transcribed to messenger RNA and is translated into a protein, which is a repressor. And it binds to the operator there on a continual basis and shuts this gene off. The promoter, the polymerase cannot get past there. It can bind to the promoter, but it can't get past the repressor. So that's just the way the normal situation. An inducible operon is switched off for the most part. So what happens now is if the cell is presented with lactose, there is a certain amount of spontaneous breakdown of lactose. And one of the products of that spontaneous breakdown, very low amounts, that's all the cell needs. One of the substances which is always associated with lactose is this little molecule called allolactose. Very similar to lactose, but it's allolactose. And allolactose is able to bind to the repressor. So what would happen is, here's the repressor here. If allolactose is present along with lactose, allolactose binds there, and it inactivates the repressor. There it is there, the repressor protein is inactivated. Now the RNA polymerase can run through and it will read off all of these genes. I will just mention that there's one gene whose name you should remember, and that is beta-galactosidase. And beta-galactosidase is the enzyme which breaks down lactose and makes it available to the cell for all sorts of other purposes. So beta-galactosidase is a really important gene. We won't discuss it in much detail, but if you go on to do cell biology, you'll learn that this testing for beta-galactosidase is one of the ways that we mark genes during genetic engineering. Okay, so that aside, the other thing that beta-galactosidase does when it produces when it starts to metabolize lactose is it produces more allolactose. So it perpetuates this whole system as long as lactose is present. When lactose is no longer present, both the lactose and the allolactose will be completely metabolized. And as soon as that happens, the repressor will return to its active form and bind here and shut the gene off. It's no longer needed if there's no lactose present. So this is our first kind. This is our inducible operon. The repressible operon, you'll see some similarities, but the repressible operon, the default is on. So this is another very famous operon. This is the trip operon. And the trip operon operates using a system where the repressor is inactive until a certain substance is present, and then it is activated and switches the gene off. So this gene runs the whole time. It's on the whole time. It runs continuously until it is stopped. So here's the trip operon. Again, we have the same structures. We have here are the structural genes here. Here's the operator there. Here's the promoter. And somewhere on the genome, not necessarily nearby, but there is a regulatory gene here, which is going to actually switch the gene on and off. So this should be familiar to you. I've mentioned this before in several guises. Remember, what we're looking at here is the trip operon. It's going to be responsible for tryptophan synthesis, not catabolism, but anabolic reaction, synthesizing tryptophan, which is an amino acid which is needed by the cell and at certain concentrations. But when its concentrations are high enough or there's no demand for tryptophan, you want to be able to switch this gene off. So this is the way it works. Again, memorize the structure and realize that here is the area of the genome which is going to be transcribed to form a repressor protein. But that repressor protein remains inactive, and the gene runs continuously. The messenger RNA is read off continuously. Here's the messenger RNA strands which end up being translated into a whole suite of enzymes which are responsible for the synthesis and transport and all sorts of other things of tryptophan. But tryptophan itself acts as what is known as a co-repressor. That is, it can bind to the repressor protein. This repressor protein normally is inactive in the repressible operon. This is inactive until tryptophan binds. Tryptophan binds to it, activates it, and it plugs into the operator there and switches the genes off. The polymerase can no longer get past. The genes are no longer read off. And this is a typical system for negative feedback pathways, because this means that what we have, but we are synthesizing tryptophan in the cell. When the concentration of tryptophan is high enough, then it switches the genes off because they're no longer necessary. When concentrations of tryptophan fall very, very low, then this repressor is inactivated and the genes are read off. Okay, so those are two methods of gene control. We must also talk a little bit about what happens in the genome if there are changes that occur in genetic sequence. And you need to realize that these changes can occur for various reasons that I'll show you in a minute. But any change in DNA sequence may have profound consequences because it may alter the protein product that's produced finally during translation. So a mutation is any permanent change in the base sequence of DNA. And we recognize three kinds of mutation. Two of them, I say, first of all, the vast majority of mutations are either neutral, they have no impact on the functioning of the cell, or they are harmful. The vast majority are either neutral or harmful. Very occasionally, a beneficial mutation may arise. It may arise and improve the functioning of the cell. It may improve the functioning of an enzyme or proteins or whatever, and give the possessor of that mutation an advantage over its competitors. So beneficial mutations occur very seldom, but they're tremendously important because this is the driver for evolution. The accumulation of beneficial mutations is what results in evolutionary change in organisms. So anything that causes a mutation is called a mutagen. And there are many kinds of mutagens, as we'll see in a while. But just off the bat, radiation, for example, is a mutagen. There are various chemicals which are mutagens. Ultraviolet light, all sorts of things can act to actually change the structure of DNA and affect the base sequence. So we will mostly discuss what are called base substitutions or point mutations. That is change in one base in a DNA sequence. Other kinds can arise. It's not the only, base substitutions are not the only mutation that can take place, but it's the most common. Most commonly, mutations arise as a point mutation. So let's have a look at the different kinds of mutations. And the first one is called a missense or nonsense mutation. Oh, sorry, a missense, not nonsense, a missense mutation. So let's have a look here. This is the template strand of our DNA. This is the normal for this particular sequence. And what we're going to look at is this here. Look at the AAA, which is going to produce a messenger RNA of UUU. So mark that there in your mind. And next to that here is C on that side and G on that side. So now let's look at a point mutation that changes that C there to a T. So in the normal, the messenger RNA that's going to be read off would be, here's our marker, UAAA. It's going to be UUU, and then normal would be G. But here, we've changed that C to a T, and now the messenger RNA reads A, UUUA. And that whole codon there now reads AGC, where before it read GGC. GGC codes for glycine, and which is the normal amino acid that should go in there. This AGC codes for serine there. What we've done is we've put in a new amino acid, and that may have serious consequences, as I'll show you at the end of our discussion. So this is a missense mutation. We've changed the meaning of the codon, and it's resulted in a change in amino acid. I should mention right off the bat that could be beneficial or it could be harmful. That change in amino acid, we don't know. We'll see this in a little while. I'll show you an example of one which is harmful. Okay, so this now is called a nonsense mutation. And this arises, again, here we are, spot your same template, right? Here's our marker for us to orient ourselves, AAA. And here's the normal sequence following that, CCG there. That's what we just discussed. We'll go a little bit up from there, go to this one here. And here the sequence reads TTC. So our messenger RNA would be AAG. And that is lysine. Here's our very first amino acid, methionine, AUG. So in a nonsense mutation, we're going to change that T there, to an A. There it is there. And that's going to produce a U. And this codon now changes from AAG, which codes for lysine, to UAG, and look up on the table of codons, and lo and behold, UAG is a stop codon. What would happen here is that this, in translation, this, whatever is being translated from this messenger RNA, would immediately stop. All you would have would be methionine. Nothing down here would be read. Why? Because a stop codon has been included. A mutation has led to a stop codon. And this is why it's called a nonsense mutation. This can be extremely deleterious, very, very dangerous. This is a very dangerous mutation. And I can't think of any way that nonsense mutations are ever beneficial or neutral. I would say they're always harmful, and probably very harmful, because you get no protein product from this gene once it's been mutated. So here is another very dangerous one. And this is when, instead of changing a base from one to another and watching the resultant change, instead, here we go, orient ourselves again, AAA, UUU. Well, now we're going to have a look at this triplet here and its resulting codon. Remember, the DNA is a triplet. Messenger RNA is a codon. Now we're going to look at that AAA triplet, and we're actually going to lose one of those bases. We're going to remove it. So what do we have now? Instead of AAA, we have AA. Here we have UU, but that was a triplet, and it will be read as a triplet, except that the triplet now will be read AAC, and it will be produce a messenger RNA codon UUG. It originally coded for phenylalanine. It now codes here for leucine. But look what happens. All of the triplets downstream are going to be shifted back. They're going to be shifted back one, and they are going to be misread. That is what a frame shift is. So that now we've shifted them back one. If I was to lose two bases here, then everything would be shifted back two, and we'd be reading completely the wrong triplets, producing completely the wrong codons. If I lose three, if I lose a whole triplet, then what we would have, we would restore the order, but we would lose one whole codon. That, in fact, may be less deleterious than the whole gene being frame shifted and being read completely wrong. So I will just mention as well that if you go to the table of codons, you will see there that many of the amino acids have several different codons which could produce them. Several different codons to produce the same amino acid during translation. If a mutation occurs in such a way that it transforms a codon into another codon, which reads for the same amino acid, that is called a neutral mutation, because although you've induced a mutation, you've changed the base sequence, you haven't changed the product. You keep the same product. Go to the table of codons and look, and you will see that for those amino acids which have many codons, there's quite a lot of variability that in the codons, which you could play with by mutating especially the last base. And those are referred to as the neutral mutations. And they have no effect on the function of the cell because they don't affect the final product. So there are many sources of mutation. As I've mentioned, chemicals. The most, unfortunately, one of the commonest ones in human beings is cigarette smoke. Weed smoke is also contains chemicals which can induce mutations in lung cells. But cigarette smoke is much more dangerous because the lung exposure to cigarette smoke is much more constant. Radiation of various kinds, electromagnetic like ultraviolet light, and especially ionizing radiation, gamma rays, X-rays, these can actually physically damage DNA. And in repairing the damage, often mistakes can be made in substituting bases that ought not to be there. There was also a lot of mutation occurs during the process of replication. But you'll remember, if you think back to what we discussed about replication, that in fact, there are proofreading mechanisms in the cell to try and limit the amount, and they are very effective at limiting the amount of mutation. They can proofread and repair errors. The last is, which we'll talk about when we talk about viruses and bacteria and how they cause disease. Sometimes mutations can arise through pathogenic means. This is especially true of viruses, and they can actually cause DNA to be altered and mutated. Okay, so I just wanted to show you this example. This is a missense mutation with its results in human beings, and this is the cause of sickle cell anemia. So here is the normal triplet CTC that is translated then GAG, and that produces the amino acid that results in putting in the amino acid glutamic acid, and that's the normal situation. So the glutamic acid is there, but all we have to do is change one base mutation, 1 point mutation in this triplet. Here, you can see CA instead of CT. CAC results in a codon GTG, and that codes for valine, not for glutamic acid, but for valine. That single amino acid change induces radical changes in the structure of hemoglobin. This is part of the gene for hemoglobin. And what hemoglobin normally does is it picks up oxygen and it transports it to the tissues. And in the tissues, it discharges its oxygen so that it provides oxygen for cellular respiration. But all of that involves shape change in the hemoglobin. When it picks up oxygen, there is a shape change in the hemoglobin. When it arrives in tissues which are low in oxygen, the shape of the hemoglobin changes and it releases its oxygen. It returns normally to its original shape. So that is the normal. You can think of the hemoglobin continually flexing inside the red blood corpuscles. This single amino acid change reduces the ability of hemoglobin to return to its normal shape. Instead, it crystallizes inside the red blood corpuscle and it contorts the red blood corpuscle into the classic sickle shape that you see in sickle cell anemia. And so these changes, this change into the sickle shape occurs in tissues in the periphery of the body, not in the lungs, but in the periphery of the body where oxygen tensions are low. And these blood cells, normally blood cells are very flexible and they can pass through the very, very fine capillaries that terminate our circulatory system and pass back and go up to the lungs. Sickle cells find it difficult to do so, and they tend to induce clotting and inflammation in muscle tissue, skin, organs, where they have discharged their oxygen and cannot return to their normal shape. So one single base change can have these very, very profound effects on the function of the DNA. Okay, so to pass on now to something else that I mentioned very briefly, I want to talk a little bit about how, in fact, DNA can be passed from one cell to another. These are very important processes in prokaryotes because genetic information, although the prokaryotes don't have sexual reproduction, which is our way of accumulating new combinations of genes. Although they replicate by binary fission, there are ways that bacteria can accumulate DNA from other bacteria. And very often that DNA that they get from other bacteria contains very useful information for them. So this, the principle of the first type of acquisition of DNA were shown by Griffith. And you probably saw this to some extent when you did the Howard Hughes Medical Institute exercises on DNA. But I'll go over it really quickly again. Griffiths was working on Streptococcus pneumoniae. And he was actually initially trying to develop a vaccine for Streptococcus pneumoniae. And he realized that there were two distinct strains of Streptococcus pneumoniae. One was called smooth, and the other one was called rough. And the reason for that is when you plate them, the smooth strain makes these glistening colonies on agar. The reason that they form these mucoid glistening colonies is because the bacteria are surrounded by a thick capsule that you can see here. Streptococcus pneumoniae usually occurs in diplococci, like this, surrounded by this thick capsule. The other strain, which is rough, do not have a capsule around them. And there, when you plate them, their colonies look rough, simply because they don't have all that mucoid stuff associated with them. The other thing about them is that this smooth is pathogenic, highly pathogenic. And if it's injected into mice, the poor mouse dies. If you inject living, rough bacteria into the mouse, nothing happens. And the reason for that is the capsule is what confers pathogenicity. It enables the bacterium to stick to cells and replicate and cause disease. The rough does not, it cannot stick, and very soon it's eliminated by the mouse's immune system. So that's the basic principle. During experimentation, Griffith came across something very strange and very anomalous. First of all, he developed methods for boiling and sterilizing cultures of smooth bacteria. So he would boil them, the cells remained intact, but they were dead. At least he could see intact cells. They didn't all remain intact, but he could see intact cells. So he knew there were cells that in the culture, but that they were non-functional. And if he injected these into a mouse, the mouse remained healthy. And he could not grow anything on a plate either, because all the cells were dead. What he then discovered was if he mixed that dead culture together with living rough culture and injected that into mice, the mice died. Why? Why would the mice die? The only living prokaryote that has been injected into them is smooth. Is rough. Is the rough version of Streptococcus. There's no living smooth to cause disease. How then does disease get caused? And even more mysteriously, how is it when he plates that he now gets smooth growing on the plate? The smooth were dead. Only the rough was living. This led to this conclusion that in actual fact, genetic information had been transferred. Somehow, he had no idea how, but initially, somehow or other, genetic information in the smooth cells was transferred to the living rough. and converted them into smooth, and gave them the ability to be pathogenic. This he called transformation, genetic transformation. He realized, of course, that there was something genetic going on. There is information contained in the smooth cells, which confers the ability to make a capsule and be pathogenic. That is lacking in rough. But when he mixes the two cultures, dead smooth, but their DNA, the genetic hereditary material is not dead. It is still there, but the cells themselves are not living. But that genetic information could be transferred to these rough cells and transform them. This is where he got the term from. It was he called it initially transforming principle, that something was transferred to the rough, which transformed them into smooth. We now understand that when you take a culture like this and you boil them, a lot of those cells are going to burst. And when they burst, they're going to liberate their DNA. The DNA will go into solution. And that DNA can be picked up by living cells. And that is the process of transformation. Transformation is the acquisition of hereditary material from the environment. And there are many bacterial cells which are able to do this. There are bits and pieces of DNA floating around all over the place under natural circumstances. And that DNA, there are certain bacteria can take that up, can actually be transformed by that DNA in solution. And the one really important way that happens is the bacteria, in addition to their genome, in addition to their chromosome rather, the large chromosome, they have these small circlets of DNA called the plasmids. And some of them have many plasmids. And the plasmids are self-replicating. They can replicate themselves. Sometimes it's tied to the replication cycle of the cell, but other times it's not. And the plasmids replicate totally independently of the rest of the genome. They're very small, only about 1.5% of the size of the chromosome, but they often contain extremely useful information. And here's our plasmids here, electron micrograph, the little circles of DNA. And here is a typical plasmid. This is called PUC19, a very famous plasmid. And this is often used in genetic engineering. So this gives you the typical structure of a plasmid. You can see here, it has a gene which confers ampicillin resistance. Many times, these plasmids contain genes for antibiotic resistance. They may also contain, as we saw for Streptococcus, they may also contain genes for pathogenicity and various other things. They can confer resistance to heavy metals. All sorts of useful information may be transferred from one bacterium to another by the process of transformation. So this is a typical little circle. This here is where its replication would begin, its own ori. And this PUC also contains a very useful gene, and you've just heard about it, called beta-galactosidase, the gene for beta-galactosidase. This is the lac gene. This is one of those genes that was in the Operon. And this is the gene for beta-galactosidase. So transformation by picking up DNA from the environment, that is one way bacteria very frequently acquire new genetic information. But there is another, and this is by conjugation. And in conjugation, plasmids are actually actively transferred from one bacteria to another in proximity to one another. They actually have to be in physical contact. And that contact is established by the production of a special tubular structure called the sex pilus. And the cells which are able to construct that sex pilus and conjugate with another cell are called F plus. The plasmid itself is called the F factor, and the cells that have it called the F plus cells. And they can copy that plasmid and donate it to another cell, convert that cell to F plus, and that cell can then go on and conjugate with other cells. So this is a typical F plasmid, but this is called also called an R factor. Those which confer resistance to antibiotics are called resistance factors. And this resistance factor is also has the genes to function as an F factor. So any cell that possesses this would be an F plus. Here, have a look here at what this gene can confer. Mercury resistance, sulfonamide, that's an antibiotic, sulfonamide resistance, streptomycin resistance there, chloramphenicol resistance there, tetracycline resistance there. And then all of these genes here are the genes that confer the ability to conjugate. So I want you to imagine that you have an infection, perhaps, rampant in a patient, right? And this patient is being treated with these antibiotics. They're being treated with chloramphenicol, perhaps, which is a very powerful antibiotic, and with tetracycline. There's a really serious infection. We're hitting it hard. Then, entering into that population comes one of these cells. One of these cells which has the resistance to all of those antibiotics that you're using, and the ability to conjugate. And when it conjugates, it's going to convert cells into cells which can themselves conjugate. Very rapidly, you can have the spread of these antibiotic resistances through the entire population. And this is one of the ways that bacteria set up resistant populations, is by transferring the information from one cell to another in the form of the plasmid by conjugation. So in order for this to happen, the plasmid has to be conjugative. It has to be able to have the genes which will enable it to create the sex pilus, duplicate the plasmid, and pass the copy to another cell. So this is the way. This is E. coli, by the way. And here is the F plus cell. This has the plasmid, which enables it to conjugate. Here it has constructed a sex pilus. This is a proteinaceous tube which connects the one cell to an F minus cell. So they don't just squirt DNA through this tube. The tube is actually to pull them close together like this and allow their cell membranes to form a pore through which the DNA will pass. And that's called a mating bridge. There it is there. What happens now is, here's the mating bridge. Here's the plasmid that we're interested in. This is the F factor, the conjugative factor. And that is replicated. It doesn't pass this plasmid from there to there. It replicates the plasmid and sends a copy to the F minus. That is fully functional. And this F minus cell is converted from F minus to F plus. This cell now gets all the benefits of those resistances or whatever information was on the plasmid, but they also acquire the ability to conjugate themselves and go find F minus cells, conjugate with them, and transfer the information. Okay, so we've got different ways of transferring DNA from cell to cell. First of all, we heard about transformation, picking it up from the environment, which bacteria very frequently do. The second way was by conjugation, by one cell adhering to another and transferring copies of the plasmid across. Here's a third way, also very important in natural populations. And this is by the process of transduction. And transduction refers specifically to the transfer of DNA from cell to cell by viruses. We haven't discussed viruses. We're going to start very soon. But let me just tell you that viruses are very common in bacteria. There are thousands upon thousands of viruses which infect bacteria. Here's one of the ways in which they work. The virus injects its genome, whatever it is, into a bacterial cell. It gets the bacterial cell to replicate the viral genome and create all the viral proteins. And the virus then assembles itself. At the same time, it chops up the genome of the host cell to render it incapable of working. And the viral particles then purely mechanically will package DNA that is present in the cytoplasm. The idea being that they are packaging their own genome that they can then go and infect new cells. But when they do this, it's relatively inaccurate. This packaging of DNA can be very inaccurate. And they end up packaging bits of the host genome. And they can then go to a cell, infect it, and instead of injecting the viral genome, they inject information from their original host. And that may be very useful information for the recipient cell. And that then gets integrated into the host, into the genome, and becomes part of the recipient's DNA. So this is a really important process in natural populations. That is transduction. Transduction refers specifically to the transfer of DNA from one cell to another by viral packaging. Okay, so I'm going to stop right there. And I think that that just about covers anything that we're going to do about DNA. And on Monday, if you want to read ahead, I'm going to very briefly be discussing epidemiology and the nature of pathogenesis. Okay, so I'll see you on Monday.